Hi, good evening. Good evening, Asraful. So, good evening. So, it says learn engineering. Okay, so you're going to do engineering, so you're not going to give NEET. Fine. So, Naveen Kumar, good evening. So, Sweta, hi, good evening. So, all those who are new to my channel, welcome. So, I am Krishnaveni. So, I am teaching biology. This is an English medium channel for weekly 3 days, that is Monday, Wednesday and Friday. I teach for class 12, and for the remaining 3 days I teach for class 11. So that is Tuesday, Thursday and Saturday I teach for class 11. So if you're new, please do hit the subscribe button, like, share. So tell your friends also there's something like this going on, so that we have a wider reach on this platform. Okay, so we are here on lecture 3. We are talking about biological classification. So it is called the sneak peek into the world of biological classification. So biological classification as such is a huge chapter, and it is pretty confusing to remember the examples, but here we are trying as much as possible to make it interesting so that you study smart and not hard, right? Okay, so let's see what I have. So today we are going to talk about the next kingdom, that is your kingdom Protista. So I know it's again a gentle reminder I've been telling you. So there's a coupon code that is KR10. So please download your eCareerPoint app. So your eCareerPoint app will have all my PPTs. So the one that I'm teaching for YouTube. So please download the app. So if at all you have to purchase any course or products, so make sure you use my coupon code, and it's valid only till 30th of November. So we're not very far. So hardly 4 days more. And also you still have only another 35 days for 2020 to get over. So I know all of you have struggled a lot, so but let's wish that the end is beautiful. Fine. So with that, let's go forward and see what I have to talk. So the agenda of your biological classification. So what are the topics that was covered? So we covered your kingdom of classification. So how to download PDF in that app. So first download the app, log in, and then you will find free courses. In free courses, go to English medium, NEET target. There you will find my classes. So go to biology. So it has both class 11 and class 12. There you will find my PPTs in PDF form. So that is how you download. Okay, Naveen? Fine. So we were talking about the kingdom of classification. So why classification was essential. Then actually we spoke about the various kingdoms. So initially you had 2 kingdom, 3 kingdom, 4 kingdom, and finally your 5 kingdom was the most validated one. So we have been talking about your 5 kingdom classification. So it has your kingdom Munira. So Munira are basically your prokaryotes. So we have been talking about kingdom Munira. So today we'll start with your kingdom Protista. So Protista consists of your unicellular eukaryotes. Fine. Then we still have to talk about kingdom Fungi. So that we'll do on your Saturday class. Then it is kingdom Plantae, kingdom Animalia. So all these were classified, and these were the 5 kingdom of classification given to you by R.H. Victator. But apart from this, certain group of organisms were neglected because they were not able to assign or assume to which group they belong to, and those were your viruses, viroids and lichens. So at the last we'll talk about them. Fine. So this was the agenda, and today we're going to talk about your kingdom Protista. Fine. So welcome to the world of Protista. So we're going to talk about unicellular eukaryotes. So kingdom Protista. So what are we going to discuss in kingdom Protista? First, we'll talk about the general features of your protistans. So how do they, what type of nutrition do they have, how do they reproduce, etc. We'll talk about their members, who are their members, then their mode of nutrition like I told you, then reproduction. So this is what we'll be talking about in kingdom Protista. And at the end, I have some questions which will summarize both my lecture 1 and 2 along with lecture 3. So that is the plan for today. That is the plan for this evening. Okay, so talking about kingdom Protista. So they are basically single-celled eukaryotes. They are placed under your kingdom Protista. They do not have well-defined boundaries because still people are confused. Okay, so whether to classify them into plant kingdom or other further classifications. So still that dilemma is there. So they are basically photosynthetic. So photosynthetic protestant or plant. So still people have a dilemma whether it belongs to protist or the plant kingdom because they are photosynthetic. So thank you, Sumit sir. Thank you so much for joining us. So the next point. So the members of Protista, the members of Protista are primarily aquatic. So this is their habitat. So remember, they are mostly aquatic. Fine. So this is your general introduction to your kingdom Protista. So now going forward, So who are the members? Who are the members under this kingdom protista? Let's see. So on what basis are they classified? So if you look at the first classification, it is based on their mode of nutrition. So whether they are autotrophic, whether they are photosynthetic. So based on the presence or absence of the photosynthetic pigment and their mode of nutrition. So autotrophic organisms, they have three members here. That is your dinoflagellates, diatoms and euglenoids. So these three come under the autotrophic protistans. So the next category is your absorptive or consumer, or they are called as your decomposer organisms. Fine. So they basically feed on your dead organic matter. They contain complex enzymes which will help in the breakdown of the complex matter into simpler ones, and that is how they make their living. So example, one member under your decomposer is your slime molds. The next one on the basis of protozoans, okay, so this is based on their movement and other special characteristics. So the third member is your protozoans. Under protozoans you have so many types. One is your zooflagellates, sarcodinoids, sporozoans and ciliates. So we'll talk about each of them in detail. So what is so unique, what is so special, and who are the examples under each category. So it's not pretty stuff. We are not going very deep. I'm strictly following your NCERT. So anything that is extra, I have put it on my slide. So make sure you note down that points. So now going forward, let's see what type of nutrition does protistans have. So I told you protistans are unicellular eukaryotes. So when they are unicellular eukaryotes, so how do they prepare their own food? And I also told you they have photosynthetic pigment. So that was our first category, right? But let's see, they again have a mix of nutrition. So it's not like they have only one type of nutrition. The first one is holophytic or photosynthetic. So from the term it is understood. So they have your chloroplast and other pigment that is essential to carry out photosynthesis, and hence their mode of nutrition is autotrophic. So that is point number one. That is the first type of nutrition. The next one is holozoic. So don't confuse between holophytic and holozoic. Holozoic is similar to the mode of nutrition in animals. So what animals do? They ingest their food, and then the food that is ingested is digested, right? First you have ingestion. Ingestion is nothing but you consume the food. So ingestion is followed by digestion. So that is your holozoic mode of nutrition. The next one is absorptive. So what does absorptive mean? So I explained you. So they're basically a decomposer organism. So they feed on dead organic substances. Once they feed on dead organic substances, they have enzymes which can break them down from your dead complex organic matter into simpler ones, and then they absorb them. So that is called as absorptive. Fine. The last one is mixotropic. It is like your cocktail. So you have both your halophytic and your holozoic. So it can intermediate between two. So that is called as mixotrophic. So this is the mode of nutrition in case of your protistans. Fine. So going forward, the next thing is reproduction. So how do the protistans reproduce? So they reproduce asexually and sexually. So both modes are possible. Fine. Asexual reproduction takes place in favorable conditions. Whenever they have a very favorable condition supporting their growth, so they undergo asexual reproduction. So asexual reproduction is of two types. One is your binary fission, the other one is fragmentation. So what is binary fission? A mother cell divides equally into two daughter cells. That is binary fission. And what is fragmentation? Okay, so spore formation. So here the spore formation is that your they will produce spores. Those spores, the spores can be produced in particular specialized structures, and the spores will germinate to produce a plant body again, a new organism again. So that is called as spore formation. So spore formation occurs in specialized structures, and that is called as sporangia, right? So spores are produced in specialized structures, and that is called as sporangia. So good evening. So sexual reproduction, so I told you they reproduce both asexually and sexually. So under sexual reproduction, so talking about sexual reproduction, it was first observed in your kingdom protista. So that is more unique here. So what happens in case of sexual reproduction? So your haploid gametes, they fuse to form your diploid zygote. So that is what happens in your sexual reproduction, and this fusion of haploid gametes is what is known as syngamy. So gamy is fusion. So this kind of fusion is called as syngamy. Fine? Okay. So going forward and talking about your syngamy, so I have to introduce you three terms, and I want you to remember them. So talking from NCERT point of view, these three types of syngamy are important. The first one is isogamy. So iso means same and gamy is fusion. So I said, what is gamete? It's a fusion. So these are the types of syngamy. So syngamy is the fusion of your haploid male and female gametes. Now, if both the type of gametes are same, they're exactly identical. They are both morphologically identical, that is in terms of structure, and physiologically also identical in terms of their genetic makeup and function. If they are similar in both the conditions, then it is called isogamy. But when they are morphologically similar but physiologically dissimilar, so what do I mean? So they are structurally similar but function wise or the genotypic wise they are different. So such a kind of isogamy is called physiological anisogamy. So anisogamy, which means they are dissimilar. Fine. The next term that I want to introduce is anisogamy. So from the term itself, it's pretty clear. So they are morphologically and physiologically dissimilar. The next one, so here you have a smaller gamete which is motile, then you have a larger gamete which is immotile. The last type of syngamy is your oogamy. So they are a developed form of your anisogamy. So here one gamete is large and immotile, the other one is small and motile. So such a kind of difference is called oogamy. So these are the three types of syngamy that I want you to remember. It is not only particular to this topic, this also is carried forward, this concept is also carried forward in your plant kingdom. So I recommend you to make a note of it, okay, because again and again we'll be talking about what is syngamy, what is isogamy, etc. Fine. Okay, so what is the life cycle of your protist? So your life cycle of protist is of two types. The first one is zygotic meiosis, and your next one is gametic meiosis. So what do I mean by zygotic meiosis? So explaining your zygotic meiosis, here your protist itself is haploid. The main body of your protist is haploid. So you have two haploid cells which will fuse and form your zygote, which is diploid, and your diploid zygote will undergo meiosis to form an adult protist. So initially the body of your protist is haploid. So two different protists come together, so they fuse to form a zygote which is diploid, and the zygote will undergo meiosis to form a adult protist. So such a kind of a life cycle is called as zygotic meiosis. So meiosis proceeds after your zygote is produced. So this is called as zygotic, and here the ploidy of your protist is haploid, so it is called as your haploid cycle. Fine, it is the haplontic life cycle, and example are your cellular slime molds. So examples are also important, so remember the examples. The next one is called as gametic meiosis. So what do I mean by gametic meiosis? So here the structure of your protist or the main body of your protist is diploid. Fine. So this will undergo meiosis to produce haploid gametes. So this is called as gametic meiosis. Through the process of meiosis, your gametes are produced. This is gametic meiosis. Here the main body itself is diploid. So it is called as a diplontic life cycle, or the predominant phase is your diploid phase, right? So here your examples are diatoms and acellular slime molds. So slime molds are also members of your protist, right? So they are basically decomposers. So when you look carefully, I gave you the example of the zygotic meiosis as cellular and your gametic meiosis as acellular. So what is the difference between acellular? So in acellular slime molds, you have the formation of plasmodium. So they are nothing but aggregates of your slime molds, that is called plasmodium. So in case of that is your acellular. In case of your cellular, you have so many cells which are grouped but they are not fused. So that is the difference between cellular and acellular slime molds. Fine. So this is the life cycle of protist. So you should remember what is gametic meiosis and what is zygotic meiosis. Okay. So now going forward, so we are going to talk about the first member of your protist. So it is called as chrysophytes. So let's see what chrysophytes have to tell us. So under chrysophytes, you have two examples. First one is your diatoms and the other one is your desmids. So di is 2. So it is like they are containing two atoms. So that is called diatoms, and the other one is desmids. So your chrysophytes are also known as the golden algae or the jewels of the sea. Fine. So let's see what more they have. So they are mostly found in your freshwater plus your marine environments. So that is the habitat that I want you to remember. Fine. Okay, so they are microscopic and they float passively in water currents. So that is, they are planktonic. So when I say planktonic, which means they are also photosynthetic, so they become the primary producers of chief producers in your aquatic ecosystem. Understood? Now, this is the most important point when I talk about diatoms. The most important point is that the cell walls form two thin overlapping shells. So it is kind of a soapbox-like structure. So it is something like this. So that is why it is called as diatom. It is made up of two cell walls. Okay. So they are overlapping and it resembles what is your soapbox. Fine. The walls of this cell, okay, so the cells of this wall, they are embedded in silica. So they are very, very hard to break. So they do not actually undergo any destruction, and naturally also it takes a very long time. So as a result, over time they get accumulated, and that accumulation is called as diatomaceous earth. The next point, so they also have ecological importance. So they help in your soil polishing, in your filtration of oils and syrups. So just remember the examples. And the most important economic importance is that they are chief producers in ocean because they are planktons. So apart from this, so these are the points from your NCERT. So apart from this, I have some extra points that I want you to remember. So not much, just few points. So talking more about your diatoms, their stored food is important, because one of your NEET question talks about a match the following, which has the members of your protistans along with their stored food. So in that case, you need to remember your stored food here is your leucosin and fat. So what is leucosin? So leucosin is nothing but a polymer of carbohydrate. So it is a linear polymer. So you can call it as a variant of your carbohydrate. So that is leucosin, and it is also fat. So that is the stored food. Then the cell wall has two layers. I told you, right? So it is something like this. So this is called as epitheca and this is called as hypotheca. So when they undergo binary fission, so one of the layers is always detained by the daughter cells. So the layer that is being retained is your epitheca. Fine. So the zygote that is formed during sexual reproduction is called as oxospore. This is very, very inclusive for your diatoms. Fine. So your examples are navicula, cyclotella and pinularia. So how to remember them? So just remember these complicated terms come under your diatom. So you can remember them as physics and chemistry. So and is how you write in terms of your WhatsApp language. So remember physics and chemistry. So if at all you look into the complicated name, remember it belongs to diatoms. Fine. Okay. So now going forward, so this is the next member. So that is your dinoflagellates. So what are dinoflagellates? So have you heard of them? So they are mostly marine and they are photosynthetic again. So your entire protistans are photosynthetic, at least the first three members, right? So your diatoms, dinoflagellates and euglenoids, but the other two, one comes under decomposers and the last ones are protozoans. So your dinoflagellates, they are mostly marine and photosynthetic. So this talks about their habitat. So you need to remember their habitat. They appear in various colors. They can appear in yellow, green, brown, blue, etc. So depending on the type of pigment that is present. So the cell wall is very stiff. It is made up of cellulose plates on the outer surface. Now hold on. We were talking about chrysophytes also. So in chrysophytes, I told you the cell wall is like a soapbox, right? So there also the cell wall is hard. Here also the cell wall is stiff. So why I am focusing on the point of cell wall is important, because in each of the member the cell wall varies. So when you talk about euglena, the cell wall is very flexible and it does not have any stiff material. Instead it is made of a protein. So you should know how to correlate and study. Again, you will have questions like this in match. So for example, the organism which has a cell wall made of silica, or the one which is embedded with silica, so you should know it is diatoms or desmids, right? So you should know how to correlate and read. So that's why this point is pretty important. So the next one, so talking about the flagella. So here I'm talking about flagella. It means your dinoflagellates are motile. So in case of your chrysophysates or your desmids or diatoms, I never spoke about flagella. So they are eflagellate, which means they are non-motile. Except during the reproductive phase, they do not have flagella. So only during the reproductive phase, they develop flagella. And then it degenerates, so they are basically non-motile. But your dinoflagellates have two flagella, so they are motile organisms. So two flagella, so one lies longitudinally and the other transversely. So remember this much. So I told you it depends on the pigment. So they can be of various colors. So your red dinoflagellate is Goldilocks. So it undergoes rapid multiplication and it produces red tides. Fine. So remember one example, that is your Goldilocks, and it is red in color because of the red pigment. Fine. Apart from that, your Glandilocks also produces toxins which are very harmful. So you would have heard of these cases if you watch a lot of Geographic and Discovery Channel. So they show you some particular cases, right? So sudden deaths when you go to the shore. So I remember seeing one episode where a child was playing on a shore. Immediately she is stuck with something. She's just playing on the shore, so a bit into the water, but not exactly or fully into the water. And suddenly she falls down unconscious. So immediately they take her back to the hospital and try to find what caused this. And eventually it becomes so severe that she's struggling for life. So you won't know these are unicellular prokaryotes. They can be harmful as well. Okay, for example, this Goldilocks, though it gives you red tides and all, it also produces a toxin. So this toxin can affect your neurons. So they are also called as neurotoxin. So here the toxin produced by a Goldilocks is satitoxin. So I think I have the name of the toxin. Okay, so again the stored food is starch here. So in case of your chrysophryates, it was your leucocin and fat. Here it is starch. Then the type of lifestyle it undergoes is your zygotic meiosis. So do you remember what is zygotic meiosis? So your protist has a haploid cell and it fuses to form a diploid. This will undergo meiosis to give you a adult protist, right? So histone proteins are absent. So mind you, we are talking about protist, and I told you protists are unicellular eukaryotes. So according to our difference between prokaryotes and eukaryotes, eukaryotes are properly packed, right? So they have what is called as histones. But this is one exception where your histone proteins are absent. So they intermediate between your prokaryotes and your eukaryotes. Thus they are known as mesokaryote. So remember this point. So apart from this, they have an osmoregulatory vacuole, and this vacuole is a non-contractile vacuole. It does not contract, but it helps to maintain the fluid balance inside the cell. Sorry. So one more point is your bioluminescence. So this is a very important point, talking in terms of your dinoflagellate. So what do I mean by bioluminescence? So luminescence is light. Bio, so when a living cell can produce light, so that is called as bioluminescence. So what happens? So when you take the protoplast of this organism, so what do I mean by protoplast? So when you remove the cell wall of this organism, the other material apart from the cell wall is called your protoplast. So the protoplast of this organism, it contains photogenic granules. It contains photogenic granules. So these photogenic granules, they have what is called as your luciferin proteins. So oxidation of these proteins will produce energy. So what happens is your oxidation. So oxidation of these proteins will produce energy in the form of light at night. So your light is now luminescent, it is visible, right? So that is the principle of your bioluminescence. So your dinoflagellates, they can bioluminescent. Fine? Is that understood? So that is one important point of your dinoflagellates. Yes, the toxins they secrete is your saxitoxin. Fine, I hope it's clear. So going forward, so the next member is your euglenoids. So what are euglenoids? So their majority are the freshwater organisms. So they are present in stagnant water. So this is the habitat. The next one, they do not have a cell wall. So remember, now correlate with the other two members. In case of diatoms, I told you you have a soapbox-like type. So it is embedded with silica and it is not destructible. In case of your dinoflagellates, again you have a stiff cellulose cell wall. But in case of your euglenoids, you do not have a cell wall. Instead of a cell wall, what compensates for the loss of a cell wall is a protein-rich layer, and that is called as a pellicle. Fine? So that pellicle makes their body very flexible. It's understood, right? If you don't have a cell wall, the rigidity is compromised. So your cell is pretty flexible. So what is the protein that is present? So that is pellicle. So remember the term pellicle. So again you have two flagella, so one is short and one is long. In case of your dinoflagellate, one was transverse and one was longitudinal. Here one is short and one is long, right? So they are photosynthetic in the presence of light, but if there is no light, so if they are deprived of the sunlight, so they do not mind, they act as predating organism. So they have the ability to shift between the type of nutrition. Fine, so they can be holozoic or holophytic. So this is a type of mixotropic nutrition. So then what about the pigments? So here the pigments are pretty identical to plants. So they have a green pigment. So your one example here is your euglena. So this is, so if you look at the organism, it's pretty clear. So they have pigments similar to your plants. So this is about your euglenoids. So now going forward, talking about your slime molds. So your slime molds are saprophytic protist. So this term is very important, saprophytic or decomposer protist. So what do I mean by saprophytic? So saprophytic means feeding on dead organic matter. So that is what saprophyte means. So the next point, so since they feed on dead organic matter, their body moves along decaying twigs and other leaves which can help them engulf the organic matter. The next one, under favorable conditions, so note the point, under favorable conditions, they form aggregations which is called as plasmodium, okay, and they grow and spread over several feet. So this plasmodium is not the one that causes malaria. So when under favorable conditions, when they are able to form aggregates, so that is called as plasmodium. Fine, this is under favorable conditions, they form plasmodium, and this plasmodium can grow and spread to several feet. The next one, under unfavorable conditions, they again form plasmodium, but now the plasmodium will differentiate and form fruiting bodies. So these fruiting bodies, they bear spores. Fine. So under favorable conditions, they form plasmodium and that grows up to several feet. But in case of unfavorable condition, your plasmodium, they differentiate into what is called as fruiting bodies. So this term is pretty important, and what happens inside the fruiting bodies is that you have the production of spores. Fine. Now each of the spore will again germinate and become a new individual. So that is how they reproduce. The next one, the spores have true walls. So that is one important point. The spores have true walls. So talking about the cell wall again, okay. So here it means that they have walls. They are extremely resistant and they can survive for so many years. So this is one member of your protist which can be extremely resistant and they can serve for a pretty long period of time on earth. And how are the spores dispersed? The spores are dispersed by air currents. So hi Priyanka, yes, true walls. Fine. The spores are dispersed by air currents. So now talking about your last member of your protistans, that is your protozoans. So here your protozoans are heterotrophs and they live as predators or parasites. So they are basically not from your autotrophic mode of nutrition. They live as predators or parasite. So predators, again, both are harmful, predators and parasites. Let's see. So they are primitive relatives of animals. So your animals are also predators, your animals are also parasites. So they are the first relative for your animals. Then you have four major groups under your protozoans. So your first one is your amoeboid protozoan. So from the name it is pretty clear. So they live in fresh water, seawater or moist soil. They have false feet. So that is what you understand from amoeboid protozoans, right? So they have false feet and they are made up of silica shells. So here the example of your amoeboid protozoan is your ant amoeba. Fine. So remember that. So let me write. So ant amoeba is your example and that is your first type of protozoan. The next one, flagellated protozoan. So from the term it should be clear, they have flagella for movement. So they are free living or parasitic. They have flagella. So that is what the name signifies. They causes diseases like sleeping sickness in trypanosoma. So the disease is called as trypanosoma. Sorry, the organism is trypanosoma and the disease that they cause is sleeping sickness. So what do I mean by flagellated protozoans? They are able to swim or they are able to move in our blood, in our circulatory system through our blood, okay? So ciliated protozoans, so your ciliated protozoans are aquatic, cilia, so they are aquatic basically, they have cilia and it has a cavity that is called gullet that opens to the outside. Fine. Yes, ciliated protozoans and sporozoans. Okay. So what are you talking about? You're talking about the type of protozoans. Fine. So one example of your ciliated protozoan is paramecium. So they have a coordinated movement, and that coordinated movement helps in engulfing of the food material. So that is your ciliated protozoan. The last one, they are called as sporozoans. So sporozoans are diverse organisms, but most essentially, the important point to note that is they have an infectious spore-like stage during their life cycle. They have that infectious spore-like stage during the life cycle, and one example which you will be able to correlate is your plasmodium. The plasmodium which causes malaria. So people often think when I say malaria that mosquitoes are the villains, but mosquitoes are just carriers. They don't know that they are carrying that plasmodium. Fine. So your plasmodium is what is responsible for your malaria. So now I have pictures of all these protozoans. So just let's quickly look into the pictures. So the first one is your entamoeba. So these are your amoeboid protozoans, and the next one, they are your flagellated ones. So this organism that I am showing here is trypanosoma. Can you look at the flagellum that is present? So they are capable of movement. The next one is your ciliated protozoans. Example is your paramecium. So if you can look properly, their body is filled with cilia, right, around their body. So that is ciliated protozoans. The last one is your plasmodium. That is your sporozoans, the plasmodium which causes malaria. Fine. So with this, we are not done talking about the members of your protist. So hope you understood. We spoke about five people. So who are those 5 people? So we spoke about your diatoms and desmids, followed by your dinoflagellates, then dinoflagellates followed by your euglenoids, then your euglenoids followed by slime molds. Then after slime molds, the last but not the least, we spoke about your protozoans, right? Yes. So thank you, Learn Engineering. So can you please give me your name? So how do I call you? So nice session. So thank you so much. Good evening. Okay. So now let's look into some questions. So this questions will actually help you cover up all the 3 lectures of biological classification. That is your lecture 1, 2 and 3. So let's see if you can quickly recapitulate what I told you. So question 1. Thank you, sir. So cyanobacteria resemble bacteria in having. So cyanobacteria was a type of member in your kingdom monirans. So monirans are basically your prokaryotes, right? So how do they resemble bacteria? In what sense? So your options are ribosomes, B, naked DNA, C, peptidoglycan cell wall, and D is all of the above. So both your cyanobacteria and bacteria fall under kingdom monera. So what is common between them? Yes, ribosomes are present in both your prokaryotes and eukaryotes. So ribosome is common. Yes, they also have a naked DNA because the DNA is not enveloped. So your DNA, the nucleus is not properly organized in case of your prokaryotes. And peptidoglycan cell wall is present in your bacteria. So your option will be all of the above. Fine. So we spoke about kingdom monera in lecture 2. So then question 2. Muramic acid is present in the cell walls of. So let me tell you what muramic acid is. So muramic acid is a amino acid sugar. So it is naturally present as N-acetyl muramic acid, and it is present in your peptidoglycan cell wall. Okay, so it is a component of your cell wall. So you should know which type of cell wall contains your muramic acid. So you should know whether it is bacteria, green algae, yeast or fungi. So yes, as Priyanka says, the answer is bacteria because your bacterial cell walls are made up of peptidoglycans, and one of the component of your peptidoglycan is your muramic acid. Fine. So that is option A. So question 3. So heterocyst is a structure which is associated with. So where did we talk about heterocyst? So heterocyst we spoke about when we were talking about your cyanobacteria. So one of the important echological point about your cyanobacteria is that they are capable of fixing nitrogen, right? So how do they fix nitrogen? So your nitrogen fixing enzyme, that is your nitrogenase enzyme, is very, very sensitive to oxygen, right? So they have this specialized structures called heterocyst which help in fixing nitrogen, and the enzyme is called nitrogenase, and the gene encoding the enzyme is called as the NIF gene. So here your option is reproduction, respiration, nitrogen fixation or locomotion. So it's a pretty direct question. So your option is C, that is nitrogen fixation. Okay. So going on talking about your question 4. So question 4, protists which are diploid, they reproduce sexually by the process of. So we spoke about the 2 life cycle of your protist, right? So one was, okay, so let me look at the options first. So one was your zygotic meiosis, cyst formation, binary fission, so gametic meiosis. So here your option B and C are not relevant, right? So you're talking about sexual reproduction and binary fission does not fit here. And then cyst formation also does not fit here. So we also spoke about your zygotic meiosis and your gametic meiosis. They're the 2 life cycle of your protist. But here they say when the protists are diploid, when the main body of your protist is diploid, so which means it is 2n. So the answer should be no, it is not zygotic meiosis. So what happens in zygotic meiosis? So your protists are haploid. They fuse to form your diploid zygote and this will undergo meiosis to form a haploid individual. So it is not one, it is not zygotic meiosis. So your protist body is diploid. So this will undergo meiosis to form haploid cells. So your answer is gametic meiosis. Fine? So this is how the questions can get tricky. Though it's a simple straightforward question, so you tend to make mistake. Okay? So the answer is D. So I have one more question. I guess this is the last question. That is question 5. Soapbox-like cell wall is characteristic of. So we just spoke about the soapbox-like cell wall. So it is a characteristic of your. So let me look into the options. Archaebacteria, diatoms, dinoflagellates, euglenoids. So just now we spoke. So the options should be easy. So the one which has your soapbox-like cell wall is. Yes, your diatoms. Yes, Priyanka, you're right. So I think this sums up the entire chapter. Okay, so I have one more question. Okay, so question 6. So decomposer protists are. So example of your decomposer protist. Yes, Surendra, you're right. So A is diatoms, B is dinoflagellates, C is slime molds, and D is euglenoid. So which one comes under your decomposer protist? So I showed you the classification of protists, right? So one was based on your autotrophic mode of nutrition, the next was saprophytic, and the last one was your predators or parasites, that is your protozoans. So one example of your decomposer protist here is your, so which was a saprophytic protist? Yes, that was your slime mold. So yes, that is the right answer. Fine. So answer is C. Yes, okay. So apart from this, I want to explain these 3 terms to you. So there's something called as facultative aerobes, obligate anaerobes, and obligate aerobes. So I want you to know the meaning and example for this facultative anaerobes. So when the term facultative comes, remember they are flexible. So they are basically aerobes, but they can also behave as anaerobes, which means they can grow both in the presence and absence of oxygen. So that is what I mean by facultative anaerobes, and one example is your E. coli. Fine. The next one is obligate anaerobes. Obligate means they are strictly anaerobes. When you give them oxygen, they will die. They can only live in environments or survive in environments which lack your oxygen. So that is called as obligate anaerobes. They are basically anaerobes and strictly anaerobes. Okay. So one example is your Clostridium species. The last definition is your obligate aerobes. Again, the same principle. They are strictly aerobes. If you take off oxygen, they will die. They strictly require oxygen for the growth. So one example is your animals and most fungi. Okay, so you will come across these terms when we talk about the further kingdom. So I thought I will explain it now. Okay, so that is how I explained you syngamy also. So remember the 3 types of syngamy, and now you'll remember the types of organisms they are, whether they are facultative anaerobes or obligate anaerobes or aerobes. So I guess we are done for today. So this has to be continued. We still have to talk about kingdom fungi. So that is remaining. So after kingdom fungi, it will be an introduction to kingdom plant kingdom and animal kingdom. Last but not the least, we'll talk about your virus, virions and lichens. So with that, we'll be done with your biological classification. Okay. So we are done for the evening. And one more important announcement. So tomorrow at 4 p.m. So I'll be teaching about teaching a chapter for class 12. So tomorrow's topic is we will solve important questions from genetics. So genetics, I mean all your Mendelian genetics. So we'll solve the questions related to your crosses, related to your molecular genetics, and also related to your evolutionary genetics. So tomorrow it's exclusive session where we are only going to solve questions from genetics. So we'll solve important questions, NEET questions from your genetics chapter. So please do join me tomorrow at 4 p.m. So I will teach you short tricks, how to find the type of gametes, how to find the genotype, etc. So please do hit the subscribe button and also set the reminder so that you get notification and you don't miss tomorrow's session. So if you genuinely like my session, please do share, subscribe and let your friends know about it. Yes, so you will come surely. So thank you, Priyanka. Fine. So this is all I want to tell you. So have a great day further. So we will meet tomorrow again. So until then, bye. Yes, Naveen, thank you. Okay, so you're waiting for my next class. I am glad you're waiting. Even I am excited for my next class. So see you all tomorrow. Bye.